All right. Good morning, colleagues. So the name is Nkosinathi Mazungula. I'll be introducing the speakers to this morning. So thank you very much for joining us in the SAIAB seminar series inside the National Aquatic Biodiversity Biobanks of South Africa. To kick us off is going to be Amanda Gura, our Biomaterials Laboratory Officer, and she'll be followed by Seshnee Reddy. So our Biobank coordinator, who is on a joint appointment between NRF-SAIAB and the Biodiversity Biobank South Africa, BBSA. So together they have over 15 years combined experience in collections and biobanking. So having begun their careers as interns at SAIAB, and they've grown through the ladder to become key figures in the biodiversity biobank space. So without further ado, I will ask Amanda to kick us off. Thank you.
Morning, everyone. My name is Amanda, as already Nati mentioned. Welcome to our talk. Today, our presentation, the topic of our presentation is Biobank Hour inside the SAIAB, NRF-SAIAB Aquatic Biodiversity.
Join us as welcome you inside the NRF.
So as a start, yeah, it's okay. As a start for our presentation today, so would like to take you through a short visual tour of our biobank, providing a brief overview of our biobank, our operations, and how to curate samples, storage, and also manage these samples.
Join us as welcome you inside the NRF-SAIAB National Aquatic Biodiversity Biobank. This biobank is situated on the first floor of National Fish Collection Building. Our biobank holds a total of 6 -80 degrees freezers and 2 -40 degrees freezers. These freezers can each store approximately 38,800 samples, which brings about over 200,000 samples. These samples are stored in cryo boxes. Each cryo box is labeled in numerical order, starting from box 1. And our boxes have 81 wells. The wells are arranged both alphabetical and numerical, starting from A to I and 1 up to 9. So all our samples are stored in 99% ethanol in 2 milliliter ethanol tubes and placed into these cryo boxes. Additionally, we also store genomic extractions. Each sample is uniquely labeled with unique box location. Just to show you how the box looks like, or how we label, and also how we label our tissues. AB23 and AB being the initials of the collector, and 23 being the year it was collected, A in the first week of that year. All our samples are catalogued on a database called Specify, which makes it easy for us to retrieve whenever we want the gifts, to prepare the gifts. The Biobank provides support to research projects by enabling access to tissue samples through these gifts. So we prepare the gift on our Biobank prep lab. That's where we do the subsampling and also the packaging. So when you are sending outside SAIAB, be it local or international institutions, we wrap them with paraffin and then place them in a padded envelope with all the necessary documentation. Thank you.
So the key components of our biobank are the samples we store. But where do we get our samples from? So we get our samples from the scientists, researchers, students, and postdocs, from you guys who are sitting here, and also the colleagues who are joining us online today. So some of you could be wondering if we are the only biobank in South Africa, and we are the largest biobank in in Africa. So the truth is we are not the only biobank. There are different types of biobank based on the biological material they collect and preserve. So just to mention a few, as you can see here, we've got plant biobanks, livestock, and aquatic biobank, which is us. The BBSA is a Biodiversity Biobank South Africa. For those of you who are not familiar with a BBSA, they're the repository of biological relevant resources, and they serve as a coordinating and supporting scientific research. They support different biobanks in South Africa. They support about 7 biobanks, which they are called core biobanks of South Africa. So their main purpose is to serve nationally and international agenda for research and development. So as you can see here, these are the 7 core biobanks within the BBSA, and SAIAB being one of those core biobanks of BBSA. So if you guys are interested in BBSA or knowing more about BBSA, we've got a website there. You can go and check what they're about. So coming back to our biobank. So what is the purpose of our biobank? So the purpose of our biobank is to serve as a repository for the storage, preservation, and management of biological samples derived from aquatic organisms. And then objectives of our biobank. So our objectives are to facilitate and support scientific research by providing well-curated facilities. We also promote collaborative and data sharing, support education and outreach on how we operate and also why we're relevant to the societal needs. I'm meeting these observations. So how do we go about in meeting these objectives? What can we do, or what are we doing, or what can we do in the near future in order for us to meet these objectives? So in order for us to meet or maintain these objectives, we need to maintain a standardized storage of our samples to ensure sample longevity and usability, which we're currently doing. And also we need a well-managed database specified in order for us to make it easy for us to retrieve the samples whenever we need to do the gifts and all that. So we've got specified, lucky for us, we have a database guru, so whenever we have a problem, it's easy for us to just go and consult. So in order for all these to run efficiently and effectively, Biobank needs to have SOPs in place that are followed. So as you can see here, we've got a list of SOPs for our biobank. So if any of you guys are interested in any or need access to any of our SOPs, you can just let us know. We'll just send it to you. Or if any of you has any suggestion on what SOPs we must add on our biobank, you are also welcome to let us know so that we can add on our biobank SOPs. Just to cover one of our main SOPs, which we think it's very important because on how to deposit samples to our biobank. As I mentioned earlier, that we got our samples from you guys. So just to go through with you guys. So in terms of compliance, you guys need to have permits and ethical clearance for your collection in order for us to accept your samples, be it tissues or specimens. That is vouchers in our language of specify. And also prepared tube labels, tissue labels and cubes. So whenever you're going to the field, especially for the guys that are inside here, you need to let us know so that we can prepare the label for you guys. So what we do, we do the labels and also supply you guys with our 2 mils append of cubes filled with 99% ethanol. So you take those with you, you just let us know how many do you need. But when you are returning from the field, you need to return all of them, irregardless of they are empty or not, to avoid the duplication of the cube labels. So you need to return all of them. You also need to have associated data for your collection, that is, be it data sheets or workbench spreadsheet, so that you have all the information for your samples. In terms of workbench, if you guys need any assistant, also Velem is your guy. You can talk to Velem, can help you with the workbench spreadsheet. So once you have all these, you can then deposit your samples to us, a biobank or collection team, because you have both tissues and also the vouchers. So obviously, if your tissue samples come together with the vouchers, the specimens, they need to be sorted first in order for us to catalog them first, so we can be able to link the tissues to them. So we depend on these vouchers to be sorted. So don't just collect and come and dump your collection to us without sorting and anything. So we also understand some of you or most of you guys, these collections are valuable or important to you guys, and you are still busy with your studies, research, you're still writing your publication. So we got you. We can put an embargo on your collection upon your request. So what we can do, we're gonna catalog all the samples, but not make them publicly available until the embargo period ends. But since, as I already mentioned, that we are a core biobank of BBSA, which we support open access. So we don't encourage you guys to put the embargo for a longer period, at least 3 years to 5 years. You must be done. Then you can extend, but they mustn't be a long period. Oh.
Okay, so continuing with how the biobank actually meets its objectives. So we're moving on to collaborative research. So this basically entails establishing partnerships. So as an example, working together with the BBSA and also the Nansen workshop and the Ocean Census workshop that was recently held at the collections. And this also includes working with you guys, so our students and our in-house researchers. And then also attending and presenting at conferences. So whether this be local or international, this is where we can showcase what the biobank has to offer, and we can also strengthen our research connections. And then we can promote and market the biobank, and this helps increase our usage, and it also helps other scientists know that they can deposit their samples into our biobank. So then when it comes to data sharing, so this, as Amanda did mention, is a very important aspect for the biobank, in that we are an open access platform. So we already publish all our specimen data on GBIF, which is the Global Biodiversity Information Facility. And then we are currently working with our postdocs and students at SAIAB to have the sequences that they have deposited onto BOLD linked onto the specified database. And then eventually the biobank also wants to publish their tissue sample data onto GGBN, which is the Global Genome Biodiversity Network. And this will be a very big step forward for the biobank. And then compliance is also a very important aspect because we share our data. So because we share our data, we have to be compliant in certain areas. So as an example, in July 2023, we were audited by the Directorate Animal Health for Section 20 biosecurity and biosafety compliance. So after a few recommendations, we received clearance on this. And then another aspect is the import and export permits that we have at collections, which are regularly updated so that we can send out specimens on loan, and we can also give out gift requests. And then, as Amanda has already mentioned, another very important aspect is that for all samples deposited into the biobank and the collections, you must have your relevant permits and ethics. So as an example, in order for us to publish our data on GGBN, we have to have permits. So if you don't have permits for that collection, it cannot be published onto that public database. Okay, and then moving on to outreach. So as we all know, it's very important for us to link our research to societal needs. And the NRF is always expressing this. So at the biobank and collections, we give tours to the students, to stakeholders, and to schools to try and help them to understand the significance of biobanking. Okay, so as with all facilities and platforms, we also run into challenges, whether this be the backlog of samples that we have at the biobank or delays in cataloging and sorting of samples. So we are making quite good progress with the backlog of samples in that we have accounted for all our samples by placing them into batches. And then our next step in this, which we are going to be working on this year, is to look at each of those batches and record all the tissue samples. So in this way, we can check whether we have data sheets in the collections for these samples and whether we have related vouchers in the collections, and then we can start cataloging our backlog. And with regard to sorting and cataloging, so when we say sorting, we are referring to the vouchers. So this means identifying them, labeling them, and rebottling them. And this is up to you guys, the collectors and the researchers. And now once this is done, the biobank will then take over, and then we will catalog your tissue samples. And then you'll have catalog numbers for your samples. And as an example, you can then use these numbers and the tissue tube numbers to perhaps sequence your samples at the AGRP. But as you can see from what I've said, this is a joint effort. So it's very important that the biobank and the researchers work together so we can avoid such delays. All right, so that was obviously a very big mouthful. So just to just collate everything. So what are the key takeaways that we can move forward with regard to this? So number one, the biobank supports and accelerates scientific research through maintaining high quality and well-managed sample facilities. And the biobank also promotes collaboration via data sharing and also adhering to strict compliance standards. But all of this is dependent on strengthening the cooperation between the biobank and our researchers, so that the biobank can have efficient workflows and also to maximize the biobank's value as a national biodiversity research institute. So from all of this, the key takeaways and everything that you've heard so far, what is my perspective as the biobank coordinator on the importance of biobanking? So I think that biobanks serve as critical infrastructure for biodiversity research by enabling access to well-preserved samples. So through efficient collaboration, curation, and outreach, the biobank can serve as a valuable tool, and it can also help in decision-making processes when it comes to the aquatic research community. And now we'll hear from Amanda as the Biomaterials officer to see what she thinks is the importance of biobanking.
I think biobank plays an important role in storing and preserving tissue samples that are potentially from rare or endangered species. The biobank also can help scientists and researchers who can't go to the field trips themselves because some of the localities are not easy access, or some of the samples were collected through expeditions. So it's not easy for one to just go and collect for themselves. And also for the future scientists to see and work with these species that might be extinct in the near future. We might seem biased since we are working in the biobank with Seshnee. So we asked our stakeholders who are working with us directly and indirectly, what are their perspectives? So the question that we asked them was, what do you think is the importance of relevance of biobanking from your experience? So let's hear what they have to say.
Hence this data. Using this data, we can look for any genetic changes within the species, and then we can then go for to make population decisions based on what we are looking at.
When I think of the biobank, I can think of these three things. First one being accessibility, that allows us to do research across the world in terms of sharing information, and when we use material from the biobank, we can collaborate. So it allows collaboration across international borders between institutes, between types of research. The second thing I think of is sustainability, because we already have reference material. We don't have to go and resample or recollect from the same places, especially when you're working with endangered species. Then the last thing I think of is the connection between the past, present, and the future. Using biobank material, you can compare your current findings with material that was compared decades ago, as well as future researchers will probably look back to the stuff that we collected, stuff that we looked at as well, and have that connection through the biobank, and being able to answer questions that probably we don't even know are questions at the moment.
Materials from the biobank can also be used for bioprospecting, where tissues or material, plant material, carbon material can be used to look for compounds with medicinal properties, let's say, for the pharmaceutical industry.
The importance of biobanking, from my experience, is that it helps us to answer certain research questions. For example, if we are looking at a species that we suspect is not a single species due to its maybe its product distribution, we can then request for tissue samples from the biobank. And then from these tissues, we can look into the DNA of the species and then see whether this the species is actually a single species or it contains multiple lineages within the species.
So the biobank is a crucial source for DNA materials, especially in cases when you're analyzing species that have become extinct, or when you're studying like a species that is far from where you are and the areas are no longer accessible. Yeah, so it's critical in that way.
Biobank is important for the long-term storage of biological material.
It can be used as access for researchers on doing research on today's specimens.
The biobank also helps in the reduction of frequent sampling in some of the critically endangered species.
When I started my career in the 70s as a fish biologist, ecologist in the field, one of the requirements we had was to have voucher specimens of the species we collected, which was quite a mission for ecologists. Because we had to bring them back to the lab, to the museum, and then accession them into the museum in case there were any queries about species identification. With the advent of biobanks, it all became a lot simpler, because then all that was required was a piece of tissue or thin clip, and the species could be identified very quickly and easily, and only a small little vial was required by the scientists. So that made a big difference, I think, to biologists and ecologists in the field in terms of lodging voucher specimens with museums using the biobank route.
So the question is, what is the relevance and importance of biobanking from my experience? Well, my experience is a long one, and biobanking and tissue preservation has come around in relatively late part of my career. However, the value of proper biobanking is absolutely inestimable. science, the science of systematics, taxonomy, of species identification, species discovery, has all been completely overhauled by tissues and molecular science and so forth, all of which requires proper documentation, proper preservation. If that proper preservation and made availability is not solid and sound, in other words, properly biobank, it all comes to zero. So biobanking, to my mind, is not only the presence, it's the future of good and solid systematics. Biobanking is something that SAIAB has done from the beginning, and it's got better and better. And all I can say is keep going. Biobanking is invaluable. It's an essential part of good science. Thank you.
So as a closing reflection to this, from what you've heard in the videos from the perspectives of others, it has highlighted the biobank as a valuable resource that supports a wide range of scientific research. And from the varied experiences that you heard about here, we can tell that biobanking is essential for the preserving of biological material that supports genetic and taxonomic studies, as well as a wide range of other studies. And the samples that are stored in biobanks can facilitate long-term research as well as enable future studies. So all in all, this reaffirms that biobanks are an invaluable tool, and that their broad relevance, they have very broad relevance and impact when it comes to species identification, biodiversity research, and conservation efforts. So lastly, on behalf of both Amanda and myself, I'd like to thank you all for attending, and thank you to our CMC team, as everything that we do is a collective effort, and a very special thank you to everyone who helped us with the videos and contributed to this presentation. Thank you so much, everyone.
All right, thank you so much. So now we are gonna give anyone who has a question to ask. So we'll start with the ones that are here, and then we'll go online.
Amanda and Seshnee, thank you very much for that presentation. This is more of a curiosity type of question. Will be subjective when you guys answer it. I don't know if you guys have seen or heard in the media, they brought back the direwolf, which has been extinct for like what, 10,000 years? Now, obviously, the work that you guys do has the potential to be used for something similar years from now. So I want to know, does that excite you guys, or does it make you guys feel worried?
It's thank you for your question. It's very exciting because the story, it's similar to our story because we have a silicon. I don't know if you know the story of silicon. It was thought to be extinct million years ago, but Ryan and the other guys are seeing the silicon still around. So we do have a tissue for it, but there's no main. I think there's 1 or 2 tissues that we have for silicon. So we'd love to have more of the tissues for that one, so that the next generation can be able to see and work with these.
Thanks for that. That's a very interesting question. So from my perspective, I do think it's like half and half. So it is exciting, but it's also a bit nerve-wracking at the same time, because yes, you are bringing back something that was extinct, but there is a reason that it became extinct, and it's also coming back into an environment that is not anything the same as to what it was before. So we don't know how it will react and what it will do. It most likely will not react the same as it would, you know, before it was extinct. I mean, but all in all, with regard to science, that's a very, very big step forward. So from what they are doing to actually clone that, what else could potentially be done if they can do something like that? So for me, that's the more exciting part with regard to the advancement in science.
All right, Roger?
Yeah, so what those guys did was they took some grey wolf DNA and they deleted or added some based on genetic evidence and fossils. So they haven't actually created the biome, but we are thinking about that because we've been in contact with people who are preserving fish material with the plan of cloning that material from somatic tissue. I think that's an invention of sperm and ovarian infection. So I mean, it's on the card, but we're a bit far away from.
Yeah. Maybe in the next talk, we need to have mics so that the people online can hear as well. Anyway, any more questions? Oh, Taryn, thank
Yeah,
you.
thanks for the talk, lads. It's always really interesting and impressive hearing what the Biobank gets up to, and you always take it for granted, I think, the role that the Biobank plays. So thank you. I must send some samples to you soon. For the open access aspect of it, you mentioned that you don't upload any of the information unless there's permitting associated with the relevant samples. What happens to those collections that may have been collected at a time where permitting and ethics and whatever wasn't, it was kind of just like okay, well, I'm sure it's fine, you can do it. So is there scope do you include those kind of historical data sets if they've been properly stored up until that point, or how do you just say that, sorry, this is the baseline and we can't do this, just out of interest?
Thank you, Taryn. Yeah, I think we do, but William's going to help on this one because he's the one who's working with the permits and ethical clearance. But I think the ones that were collected many years ago, I think they are covered. I think this thing started recently, maybe in the 2012 or 2010. So that's where we were more concerned about those ones, but the old ones, I think are covered.
Yeah, so I think for GGBN specifically, it's after 2014.
Okay.
So before
So they've got a base,
2014,
yeah.
you can still put samples on that don't have permitting, because as you mentioned, there's a lot of samples in the biobank that at that time that they didn't need to have permits for ethical clearance. So it's more for our recent collections that are happening now, because those are already in place. You have to have permits and you have to have ethics.
Just to add on that, so we have not published anything now. So we have not published any of our TCs on GGBN. So that's what Willem is working on to make sure that whatever we're going to publish has all the relevant permits and Francesco.
Thanks a lot. Now you're taking tissues and you don't need so much space, but how about, for example, I think one of the big potential, and you listed of biobank or collections, is access to historic material. And a lot of change can be measured, and I'm not a fish expert, but if you preserve otoliths. So how about potential expansion in otoliths collection, which really could, and it's not only fish, invertebrates also, whatever has got a ring of growth has got that potential. How about considering that? And also, you want samples, you want to work with researchers. A lot of us go onto the field on a monthly basis. How much capacity or storage capacity do you have to potentially engage into that almost like bulk, which would give great information on timelines and time change. So these two little additions.
Thanks, Francesca. Again, the storage is not a problem
for now.
For now,
It depends on the tree end.
Yes, it depends. So we can think about having to store some otoliths and stuff. So
yeah, we are looking at expanding the biobank, and you're right in that we need to expand it with regard to the research interests. And as you said, the research is here, that's where we can start. So if otoliths is something that's very big, if stable isotope analysis is something that's very big, then why not start that collection? Because we know that it's something that's going to be used. So the main thing also is that we don't want to collect samples and it's not being used, because the whole point of the biobank is to also for our samples to be used. So yeah, we're definitely open to more than just storing tissue samples and genomic DNA. We've got lots of
We will start with leadership.
Yes, thank you very much, Amanda and Seshnee, for the wonderful job. I really understand now what I is. But you would appreciate that the perspective of research now is shifting slowly but surely into commercialization. Are there any ways of commercializing what you guys are anyway, you understand what I'm trying to say. Do you have any plans of doing such that English way?
So commercializing
Yeah.
the biobank and how we give out our samples.
Yeah.
So at the moment, and I can be corrected if I'm wrong here, but at the moment, the biobank only supplies samples for research purposes. But we were in conversation with regard to potentially sending our samples out for commercial purposes as well, but we haven't reached that stage yet. I think that it will be a matter of looking at the compliance as well and seeing what other documents we may need in order to make that happen. But I don't see, it's not something that I would say, no, that's definitely not going to happen. It's not happening now, but it can definitely happen in the future with just some adjustments to our operations at the biobank.
Okay. For me, very easy. The correlations between the use and the collecting is the data that says that it's a good sign that people are using this biobank, instead of having a biobank that is not like the efficiency of the output in terms of research. Talk on that?
Oh yeah, our biobank is being used, but not as the essence of the collection. But we have guys inside here, SAIAB, the likes of Fionella, they've been using our biobank, publishing all the new specimens and all that. So yeah, it is being used even by the people outside. SAIAB, as I've mentioned, we're sending gifts left, right, and center. So we are being used.
Yeah, I want to add on that. The biobank is being used. Each week we are sending out gifts, left, right, and center. A gift. So it's a gift because we are giving you a tissue, you're not gonna give it to us back. So we are losing that. But then we require that you sometimes to let us know where the sequence, so that we can link it. And also sometimes because I see people now with the requests that have been coming saying, can you please make your piece bigger?
Yeah.
But then it's depleting now, because when people go out to the field, they don't take a big piece, it's a small piece. So it's gonna get depleted over time. Can you imagine if you're taking CO1 now, another person is gonna come and ask to do a CO1 on the same thing. So I wondered maybe if you can touch on what does the biobank do now, because these things are gonna be depleted. Maybe some of the projects, if you can touch on the whole genome sequencing.
Yeah. So one of the things that we are working on with regard to depletion of our samples, that as Nkosinathi mentioned, when we send out a gift and once the sequencing is done and it's made publicly available, so it's on Bold or GBIF or something like that, we would like to have that information. So even though we don't have the tissue sample, we at least have the sequence. So we have something that's related to that, so we don't completely lose it. And with the funding that we have received from the BBSA as internal with the biobank, we are also working on sequencing some of our samples and also getting whole genomes, which is really good for us because in the past, we always depended on the researchers for this purpose. But now we can actually select samples that we think are important in the biobank and we can get that extra information as well. And then Thabo, with regard to your specific question, we also record all the gift requests that we get, and that's all always logged in the management meetings and things like that. So from my opinion, I do think that the input that we get is matching the output.
And also when people publish, they acknowledge us and then send us those. And some of you, many of you here graduated using the Biobank. So I think it's a good thing, yeah. Other questions? There's only one question. Oh, there is Martin Jordan online. Okay, you can ask. Okay, so Martin, you can ask.
Hi, thanks everyone for a really good presentation. It was very I really enjoyed it and learned a lot. I just have a quick question regarding the mention of permits. I work for Cape Nature, the Provincial Conservation Agency, and based on our Provincial Conservation Ordinance, we are allowed to collect biological samples. So in terms of the permit and ethics requirements for logging samples, would we as conservation officials be covered by the ordinance, or is there any additional documentation that we would need to be able to contribute to the collection? Thanks.
Yeah, I need to mute you. Yeah, I think the one who can answer that because they don't have ethics approval, and we want anything that's coming here to have an ethics approval. So Willem is gonna come and address that. Chairman of the Animal Ethics Committee.
Yeah, thank you. I think it's very important for SAIAB scientists to work with any external parties that need to lodge specimens in the biobank. That is the secret to getting ethics approval. I think we cannot accept a single tissue sample that is not accompanied by an approved ethics protocol. So it's very important for somebody outside SAIAB to partner with a SAIAB scientist and develop that ethics protocol and have it approved.
So maybe
sorry, just as a follow-up to that, then would it then be an option for us from the conservation agencies to actually, when we do research and we could potentially contribute to run the ethics through the SAIAB ethics committee?
Yeah, as I say, preferably to actually have a SAIAB collaborator to work with you.
Okay.
As a co-author of the protocol, the ethics protocol, yeah.
Okay, and sorry, sorry, I'm dominating the conversation a bit here. Would the ethics proposal then be sort of like project to project, or would there be something like if we could say what our annual work plan is, that we could say we're sampling areas X, Y, and Z, these are the species we're hoping to target, can you then get ethics application and subsequent approval for maybe on an annual basis covering different projects, or would it have to be project per project?
Yeah, it is project-based, but if you word your protocol to be very broad, if you just you're a bit clever with the way you do it, I guess you can be more efficient. But yes, strictly speaking, ethics protocols should be project by project, definitely.
Okay, thank you.
All right. So we have another question. Thank you. So we have a question online. Thank you very much for the presentation. It was very informative. As a Rhodes MSc student conducting research remotely on abalone farm, I occasionally encounter species that neither I nor my supervisor can identify. Can you please outline the protocols for preserving such unidentified species for submission to the biobank? Additionally, are there any specific requirements or restrictions for researchers who wish to deposit samples in the NRF-SAIAB biobank? I think we've answered. Yeah, I think we've covered that already. All right, another question. Good morning, colleagues. I just want to clarify if the section 20 you mentioned earlier is specifically for research study provided by a person conducting the study, or does the biobank has its own section 20 permit? Yes, the biobank has its own section 20 permit to operate. Roger, you had a question?
Yeah, it's relating to what Martine said. So a lot of what Martine is doing is collecting voucher material, and I believe that would fit within the REFET program, of which I'm sure she's a co-researcher.
That's something that we can discuss together. One of the things that I want people here to understand who go out to the field, you can take a tissue sample and deposit it to the biobank without the fish. We have a fish observation. So I was in a meeting yesterday. There was an excitement about some record in the Kawi, and that information doesn't go to the collection. But if you encounter something exciting, even if you don't bring it to us, you bring a picture, we catalog it, so that we know that the record is specified, because no one will know about your exciting record. It will forever be in your phone. So you need to share that information, because someone else might want to know. So when you go out, you can share pictures, we can catalog them, as well as, tissues with pictures. You don't really need to bring fish as well, but as though we would want fish as well. So, but please let us work together on that one. All right, no more questions? I'm sweating here.
Sorry, it might be useful for everyone here and people online to maybe just share what, at the bare minimum, would it be useful if you go in the field, say you're collecting a fin clip, for example, would it be useful to take a photo of the animal? Do you need associated metadata? How big is it? Where was it caught? Location, all that kind of thing. So what at the very least would be helpful information to bring back with your sample?
Yeah, so it's fine. So as I already mentioned on my presentation, on our presentation, so you need a picture for that one. Also, the information, the metadata will need to fill in the data sheet or the spreadsheet, as I mentioned. So we need all that, even if it's just a tissue. So we need all the information where it was collected and stuff like that, the measurements and yeah.
The more data, the more valuable is
the better, yeah.
But I mean, as a minimum, obviously a species identification, and that can be linked to the photo, and then locality information, which I think is something that when you go out in the field, you already would take that information anyway. So that at a bare minimum would be species identification and at least coordinates and location.
Is it so? Thank you very much for watching.